Well, good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Environmental Monitoring, Evaluating Cellular Response Pathways in Complex Mixtures. My name is Ryan Muse, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speaker. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, please contact me at any time by sending a message using this same chat panel. At this time, all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Indigo Biosciences, who developed the content for this presentation. Indigo Biosciences is a leading provider of cell-based luciferase reporter assays. They offer assays as turnkey kits for use by scientists in their own labs, or Indigo can perform the assays as a service. Their products and services are used in a broad range of industries, such as environmental, drug discovery, academia, and contract research organizations. Their assays have been demonstrated to provide fast, accurate, reproducible results, and Indigo is dedicated to supporting scientists through reliable science, easy-to-use products, and highly qualified technical support team. Now I would like to introduce our speaker for today's event. Dr. Van den Heuvel is a recognized expert in the field of nuclear receptor biology and toxicology with over 100 peer-reviewed publications. In addition to his role as Chief Scientific Officer at Indigo Biosciences, Dr. Van den Heuvel is a professor at Penn State University, where he is program coordinator of the undergraduate pharmacology and toxicology program and leads an extramural-funded research program. But now, without further ado, I'd like to go ahead and hand things over to our speaker for today. Dr. Van den Heuvel, you may begin when you're ready.
Well, thank you, Ryan, and welcome everyone. So today we're going to be talking about tools and approaches for examining environmental quality. In particular, we're going to be talking about complex mixtures of chemicals that are found in surface water and drinking water. So what we're going to do is we're going to start out talking about what the problem is, what the main types of problems that we are seeing in the environment and problems to doing biomonitoring. And then we're going to talk about two major approaches for examining water quality. One we're going to call the EAR approach, and the other is going to be using some bioassay systems to examine activity within water samples. And then during the conclusions, I'll compare and contrast these two different approaches. So what's the problem that we're trying to discuss here? A lot of what I'm going to be discussing at the beginning of this talk, as well as at the beginning of the biomonitoring section, has been covered in a previous X talk called Biomonitoring Relevance of Bioassays, which you can find by going to the X Talks website and just search for Indigo. And so the problem that we're addressing here is trying to examine water quality. And I think this is a very common problem across many government agencies, as well as academic labs, et cetera. And we have a lot of tools at our disposal and a lot of parameters that we need to meet in order to make decisions about what remediation needs to be done, what are potential sites that need action. And so we have a variety of different parameters that we look at to examine the water. And what we're going to be focusing on are what I'll term micropollutants, or chemicals of emerging concern. And so these are going to include pesticides, pharmaceutical, personal care products. These are found in water, but we don't have safety levels set. Whereas some of these other parameters, we have clear numbers, clear values that will determine what action needs to be made. So it's really these micropollutants that are a problem, mainly because we have so little information on them. So we're gonna call these micropollutants contaminants of emerging concern. They're emerging because they may be newly identified, newly manufactured chemicals, chemicals that are put into the market that are finding their way into environmental matrices. And as I mentioned before, in many cases, these micropollutants do not have regulatory health standards. We know they're there, but we have no action levels present for these compounds. But they are found in a variety of different matrices, groundwater, surface water, wastewater, et cetera. And many of these compounds, in addition to being have very little information about them, what we do know is concerning. Things like their persistence in the environment, their ability to accumulate in the food chain, but again, in the absence of real information about what risk this may pose. And another additional problem is we're never talking about an individual chemical at a time. We're talking about the ability of these chemicals to interact with each other to cause additive, synergistic, and antagonistic effects within this environmental sample. So what we want to do, and what our approaches that we're going to discuss today, what we're trying to do is look at a particular study. And we're trying to prioritize sites, chemicals, and bioactivities that may be of concern. So right now, the majority of the approaches that are used to examine water quality are simply the analytical approach. So trying to predict what types of chemicals are there, or have a concern over specific types of chemicals, collect the water samples, extract, and then use the classic analytical chemistry approach and simply get concentrations. But as we'll talk about, concentrations of chemicals, presence of chemicals is a determinant of activity, but it is not telling you the overall water quality. So what we're going to talk about today are two different approaches for trying to address the mechanisms by which these chemicals cause toxicity and the ability of these chemicals to work in concert with each other to cause an adverse event. So the first is called the EAR approach, and this is exposure activity ratios. And this is where we use the existing concentration and then compare this to known effects, known dose response relationships that are present within a database such as the ToxCast database. Then we're going to contrast this to actually measuring the activity of these samples against a variety of different targets that we know are involved in an adverse outcome. And we'll define these as molecular initiating events that are key steps in actually causing a biological response in a cell. So again, we're going to go through these two different approaches as a means to try to prioritize the sites, prioritize the chemicals that are present, and to try to address any potential bioactivities that may lead to an adverse outcome, either in humans or in wildlife. And both of these approaches really embrace trying to understand how chemicals result in an adverse outcome. And this is called an adverse outcome pathway or a toxicity pathway. And this is a series of linked events that are tied to leading to some disease or impaired development and reproduction. And this, again, there's a series of interwoven events that is initiated by a key event called a molecular initiating event. And this is where the chemical is interacting with some cellular macromolecule that starts a chain of events that leads to what are known as key events, things like altered gene expression, altered second messengers, changes in cell-cell interactions, which all of these key events tie together to lead to an effect at the cell level, an effect at the tissue level, effect at the organism level, and perhaps an effect at a population level. So we're going to be thinking about meeting our goals of trying to address water quality using as much information as we can about these steps in an adverse outcome pathway. Because remember, this is going to be predictive. So even that first molecular initiating event is going to be predictive of a more complex adverse outcome. And there are a few of these adverse outcome pathways that are important for addressing compounds of emerging concern. The one that most people identify with is shown at the top here, which is endocrine disruption. So this is a chemical that is interfering with the reproductive effects, with proliferation, maybe affecting cancer within humans and susceptible wildlife populations. This adverse outcome has several molecular initiating events that are very important, and the types of molecular initiating events that are involved here are nuclear receptors, such as those that respond to the androgen and estrogen and thyroid hormone. So these are the three key targets to leading to endocrine disruption. Something that has been less utilized or less realized is that there are other ways of disrupting endocrine function, and that is mainly by altering the internal levels of these natural hormones. And this may be done by what I'm showing here as metabolic disruption. So your compound of emerging concern may interact with receptors like the pregnine X receptor or the constitutive androstenedione receptor, regulate gene expression. These altered levels of different enzymes then are going to affect the levels of androgens and estrogens and thyroid hormone within the organism, and ultimately is going to lead to endocrine disruption as well. So we are going to be discussing many of these different molecular initiating events. These all fit into a family of nuclear receptors or transcription factors that are responsive to a variety of different compounds of emerging concern. All right, so now that we have a basic understanding of what the problem is, a basic realization that there are key molecular initiating events that are tied to these adverse outcomes, let's talk about two different approaches to address water quality, taking these into account. And the first is the EAR approach, which stands for exposure activity relationship. And as I mentioned previously, this is a very simple concept where you are looking at the concentration of the chemical within your environmental matrix, and you're relating that to the ability of this chemical to cause an effect. And this effect that we're looking at could be a key event, it could be a molecular initiating event, it could be a cellular response. And the way that we're looking at the dose response effect of this chemical to cause an effect is we're using a measure of potency, which is called the AC50 value. So this would be for a particular pathway, at what concentration do we get 50% of the maximal response? And this could be an increase in activity, so an EC50. It could be an IC50, an inhibitor concentration 50% as well. So what we're doing in this case is we're evaluating the concentration of an individual chemical against its potency. And how we're doing this is comparing this chemical to a database on the ComTox dashboard called ToxCast, where they have thousands of different chemicals that have been evaluated across a variety of different endpoints where we have AC50 values. So this EAR is very similar to a hazard quotient for those of you that do risk assessment. So this is just a ratio of the concentration relative to the potency. Higher concentration, higher EAR, lower AC50 value, lower potency, higher EAR. And the way that we have used that in this particular illustration is we've used a open source package called ToxEval, which simply takes your chemical, pulls all the AC50 values in certain pathways that are present in the ToxCast database. And how you interpret this data is really combining all the different EAR values for each of the pathways and summing them to get a total exposure activity ratio for a chemical. So a chemical may have multiple endpoints where it has an EAR value. You simply sum those up for the chemical. You may look at a pathway and say, well, how many chemicals are interfering with that pathway? And you simply add up all the chemicals that their EAR for that pathway and sum them together. You can do this for a site where you take all of the EAR values for each chemical at each site and sum these all together. And so this is a way of looking at the relative quality of each sample using a very easy to evaluate ratio. But remember that this is looking for the potential for biological effect, and it's relying on the fact that this data is in the database and that all the data is well managed and well curated. So let's use an example. In this example, we are going to utilize for both the EAR approach as well as for the bioassay. And I thank the collaborators here at Penn State and in the USDA for providing these water samples to us. So these are our surface water samples from Center and Huntington counties here in central Pennsylvania. These are impacted by agricultural activity and private septic systems. But we are concerned that there are chemicals that are present that may be causing some adverse outcomes. Water quality in some of these streams is of concern. So our collaborators have examined 48 chemicals. Again, these are mainly pharmaceuticals, personal care products, and pesticides. Of those 48, 43 were detectable in these samples. But out of that 43, only 33 had data that we could interrogate, that we could actually calculate EAR values for. And I'm showing all the chemical names that are present. All right. In most cases, the first step in doing this environmental studies is simply just getting the concentration of the chemicals within each of the water sites. I should have mentioned that we actually have 9 different environmental sites that we've picked, and we have 1 blank. And so I've listed these simply as sites. And you can look at these and ask, how many sites do we see these chemicals present? And then you can look at what is the concentration average. So in this graph and some of the other graphs that I'll show you, these box plots simply show the high and the low value. And notice that this is also on a log scale. So we have very different levels of concentrations at different sites. We have quite a wide range. We have some that are only found at 1 site, some that are found in all 10. But again, this is simply their presence and their concentration. It is not telling you about the risk. So now if we take this data and convert it into EARs, and remember, each of these chemicals may have multiple pathways that they are interacting with. So one compound, let's say DEET, may have 5 different pathways that it interacts with. We are taking the EAR for each of those pathways, combining them together. Again, this box plot is showing at which sites did we see, were we able to measure EAR activity, and then the high to the low range. A couple things to point out here is that we have several chemicals that, again, looking at the EAR value moving from left to right means that we have a higher amount of concern, higher exposure activity relationships. We have compounds Shown here, the pharmaceuticals at the bottom, pesticides are in red, and personal care products are in blue. We look at this, you can see that carbaryl and permethrin have high EAR values, but you'll also see that we have gaps. So we were able to detect these compounds at the bottom here. They're present in more than one site, but there's no ToxCast data, so we cannot get EAR values. But we can use this data to start to prioritize, so we can get a list of the priority compounds, which ones have the highest EAR values are going to be of the most concern. We can look at what class of chemical is of most concern. In our case, it appears to be the pharmaceuticals and the pesticides, less so with the personal care products. We can start to look at the different sites and try to say, well, are there different sites that have different components that are contributing to the EAR value? And then on the far right here, we can say, well, what pathways are affected by individual chemicals, and later on I'll show you an example where we actually pooled multiple chemicals together. In this case, with the compound that had the highest EAR value, you can see we're getting the majority of the activity is actually coming from androgen receptor, but we're also seeing things like FXR, PXR, and the estrogen receptor are also being impacted. So what are the conclusions from this set of studies? Is, well, first of all, we do know that many of the chemicals that we are interested in are found within these water samples. In data that I'm not going to show you, but will be coming out in several manuscripts, is that there are site and time dependent differences in these chemicals. And we are also addressing stream quality to try to compare the presence of the chemicals to the overall quality at that site. There are several chemicals, however, that don't have sufficient data to generate EAR values, so they were blind to their effects. We do know that pharmaceuticals and pesticides seem to be those class of compounds that are of most concern. This carbamazepam is the most prominent pharmaceutical that is of concern, whereas pesticides, there were several instances of compounds with high EARs, such as permethrin and carbaryl. There were pathways that tended to show up with these chemicals. Many of these are involved in endocrine disruption, such as AR, ER, and thyroid hormone receptor. So we are seeing some of our concerning adverse outcome pathways may be impacted based on the presence of the compounds and their known effects. But remember, everything that we've done so far is using data that is present within the ToxCast database. There's gaps. Now let's use a different approach, the bioassay approach. And what I mean by bioassay, and again, this is something that we talked about in the previous X talk, bioassays are just simply simple test systems to evaluate whether a compound or an extract has a particular biological response. And so these are driven towards sensitivity and robustness for particular endpoints. And these can include changes in gene expression, changes in metabolites. We quite often use our endpoint looking at altered reporter gene activity, something like luciferase activity. There are some bioassays that rely on higher levels of biological activity. These give the ability to look at more complicated effects, but from a biomonitoring standpoint, they are too complicated to use to a high degree. What we're going to be focusing on is really molecular initiating events that are tied to an adverse outcome pathway. We can use these molecular initiating events to look at mixtures, and we can also use higher throughput screening processes. Many of these bioassays that we're going to talk about for looking at complex mixtures are the same that are used in the ToxCast program to generate the individual chemical data. But remember, this is not looking at theoretical activity. This isn't looking at an ear. This is actually looking at actual activity within that sample. So if you were to address that sample set that I mentioned before, that we ran the ear approach on, where we had 80 samples, this represented 10 different sites at 8 individual times, how would you address the biological activity in that? Well, one approach, and I'll come back to this later on, is simply measure everything. So take all 80 of these samples, run every molecular initiating event that you think may be at play, or try to discover new pathways that you hadn't anticipated. So many people would approach this saying, well, I know that many chemicals are going to interact with estrogen receptor. So therefore, that's the bioassay that I'm going to choose. There may be mandates for certain assays within a government agency where they are mandated to look at AHR activity, estrogen receptor activity, and androgen receptor activity. So in those cases, yes, you definitely would use these bioassays. You'd look at your mixtures and assess, has your compound led to a certain level of activity? What I'm going to talk about today is more of a bioassay discovery and something a little more open-ended. You're interested in overall quality, not just interested in whether it is interacting with 1 or 2 receptors. One of the problems, if you were to go through and just look at all of these different receptors, all the different assays that are available, is in actuality, you're not going to get activity of all of them. In fact, with this particular data set, with these 80 different water samples, we found that basically only 5 appeared to have activity in the majority of the samples. Which is highlighted with those red brackets. The green brackets, we might see a little bit of activity here and there, but it's not consistent across site, across time points. So basically, you have run many assays and generated very little data. So one of the approaches that I'm going to talk about now is some way to try to get the equivalent data to what's shown here, but without running the 30+ individual assays. We've actually run around 50 different assays on this particular sample set. So the approach that we've used, and we've published this a couple of times, and we have a couple of publications in the works, is utilizing a pooled approach for these bioassays. So we're trying to take multiple receptor assays, put them into a single tube, and then add your compound, your extract, and ask whether you have activity or not. So we've tried to streamline this. We've tried to make it more practical to try to, again, winnow down the number of bioassays that are necessary to evaluate your particular sample set. So the way that we have come to look at this is to go through the general water sample and extraction as we've always been doing, and then try to, instead of looking at each extract and doing a dose response, we have done a cytotoxicity dose response and created a maximum tolerated dose library. And now this is the library that we're going to do our screen. So we're not going to be looking at extracts that are causing cytotoxicity that may be masking or confounding the data that we get. So we take our library of extracts, and we first take a subset of our overall library. In the case that I'll show you on the next slide, we're taking 10 out of the 80 samples. We picked one time point, the 10 different sites as our sub-screening subset. We took that subset and we evaluated each of the different panels that are shown here. So we have 6 receptors that are involved in xenobiotic and biometabolism, 6 receptors, lipid and energy metabolism, endocrine and reproduction, basal metabolism and CNS, and toxicology and inflammation. Took our screening set and asked the question, did any of these samples activate something within each of these panels? If a panel is positive, then we'd come back and ask which of those receptors that are present in that panel showed the activity. In this way, we were able to get down to, instead of looking at 30 different receptors, we were able to get down to a handful that then we can use for the full screen and know that these have the highest amount of hits within our data set. So let's walk through some real world data with this. So again, this is the same sample set that we used for ear, 9 different sites in Center and Huntington County, 8 different sampling dates. So here's our creating our maximum tolerated dose. So on the left, this panel is showing the toxicity of the original extract, and then the toxicity after we diluted it to the appropriate amount. And some of these samples required more dilution than others to get them to be to lead to greater than 80% viability. We took that one time point as our representative samples, and we examined each one of these sites, extracts in our different panels, and simply asked the question, did we get activity in one of our samples? And you can see here we saw activity in our xenobiotic metabolism, our endocrine disruption, and our basal metabolism. So we looked at those 3 panels, took the individual receptors, we ignored the lipid and energy metabolism, and we ignored the toxicity, and then looked at each of the individual receptors. So the xenobiotic metabolism had AHR, CAR2, CAR3, FXR, PXR, and VDR, showed that our activity is coming from AHR and PXR, and so on with the different different panels. We were able to determine that there are 5 receptors where we're getting activity. And now we can go back, and with these 5 receptors, we can look at our biological activity across sites, across time, and we can start to now evaluate quality using our most responsive assays. So using this approach, we were able to cut down on the number of assays that we needed to perform. We were able to quickly prioritize which pathways are of most importance for this particular sample set. So what did we gain from this? Well, certainly money and time. So as what's shown down here, instead of running 20, 29 or 30 assays, we are receiving basically the equivalent data with just our 5 assays. But we also had other things that I think we needed to prove with this data set. Again, with this data set, we measured everything, and then we did this streamlined approach and compared to see what we missed. So one thing that we do see is that basically by looking at each individual receptor, that if we had something that did hit in our representative panel, our first group of 10, it tended to hit in the full library. So picking a sampling does predict what's going to happen in the full data set. If a panel is positive, we would always find that there was a positive single receptor. If it was negative, this was truly negative. We did not find any individual receptor that did activate if our panel showed to be inactive. But what did we lose by using this approach as opposed to just running each individual receptor? Well, the first thing is shown here, the sensitivity is not the same, which makes sense because what you're doing is in effect diluting each receptor in sixth because you're pooling 6 different receptors into one single assay well. So we could detect differences in the panel, but they tended to have a lower maximum RLU, and we tended to have a higher limit of detection. Really can't do this for all types of molecular initiating events. For example, there are many important biological responses that aren't agonism responses. So antagonistic responses, as well as inverse agonist response, which is shown over on the right panel. So there are several receptors that are known to interact with environmental chemicals that, instead of leading to an increase in activity, actually lead to a decrease in activity. The estrogen receptor related receptors, alpha, beta, and gamma, as well as retinoic orphan receptor, ROR gamma, are shown on the right. We couldn't interrogate those using this approach, but you can see that they are being activated by a variety of different samples within this data set. A couple other things to note is by using this small subset, you may lose things that are happening in the whole library as a whole. So looking at our sample set, which is highlighted, you can see here that some of the activities and some of the chemicals were not present within that initial data set, that subset. And so we may miss things later on. Another thing that I would say is that using this maximum tolerated dose, we are erring on the side of not having toxicity to try to make sure we're not interfering with the other endpoints. But this tends to skew things where we are not seeing some of the toxic responses, some of the inflammatory responses, and things like the DNA damage response, P53. All right, so conclusions from this part of the talk is bioassays are an impactful way of looking at matrix quality. We can easily say, yes, we have this type of activity within the sample. We can prioritize sites. We can answer whether a remediation effort has removed that biological activity. You don't need to know that it removed specific chemicals. You just know that it removed the bioactivity. These are sensitive. We can measure, depending on the assay, very low levels of environmental compounds. We can also look at the cumulative effect. Any chemical that is interacting with this molecular initiating event, we can interrogate. One of the things that we're finding, and one of the things that I'll mention later on with the data that comes from the ToxCast data set, is this is data that is in the site that is taken over years and years, and some of the data may be of different quality than others, may be done on different platforms, may have slightly different endpoints than some other assays, even though they're being utilized in this EAR approach. So this data is in a single type of platform. We know how they act with these samples. We know the limitations of each different assay. And also, this is real activity, not virtual. So this is not predicted activity. This is actually what's measured in that sample. And again, this is embracing the mechanisms. So we are looking at specific adverse outcomes, and particularly we're looking at molecular initiating events that are involved in endocrine and metabolic disruption. Some of the benefits of this approach, in particular, compared to just being mandated which receptors you're going to look at is that this may identify pathways that you may not have predicted. You may not have noticed on the previous set of slides, but we had mineral corticoid receptor activation as a surprise in this data set. This wasn't what you commonly think of when you're looking at environmental samples. You generally think of AHR, PXR, androgen, and estrogen. Well, those were present, but we would not have predicted MR activity. I alluded to this previously, but this approach using bioassays, you can use a variety of different environmental matrices, not just water samples. Basically anything that you can extract and get into a solvent that is compatible with cell-based bioassays, DMSO is the preferred solvent. Anything that you can get into DMSO, we can use this approach and look for biological activity. So this could be, water, sediment, soil. It could be silicone wristbands. It could be air samples that have gone through a filter. All right, so for conclusions, what I'd like to do is kind of compare and contrast both of these approaches. Again, the goal is to look at environmental quality, be able to prioritize sites, chemicals, and biological activity. So the EAR approach, if we look at the site prediction, you can see that it's predicting that several sites have high EAR values, whereas sites 5 and 6 are basically at the level of concern as the blank. It also determined that carbamazine is the most concerning chemical. When you look at the data, you realize that a lot of the EAR values for this chemical actually come from one assay, or from one AR assay that is driving that particular EAR quantification. And it just turns out that sites 5 and 6 are low in this chemical. That's why the EAR value is low. Whereas if we did this approach with the bioassay and look at the sites, I think you can see a clear difference in the predicted site quality. In this case, our sites 1, 2, and 3 are much different than our other sites. Our blank site is as you would hope, is very low. So looking at these 5 receptors and pooling their bioequivalent quotients, you can see that site 5 probably has the most biological activity, and this is predominantly due to high androgen receptor activity. As I mentioned, the MR, mineral corticoid receptor activation in these environmental samples is a blind spot in the EAR approach. It is actually, there's not a lot of mineral corticoid receptor data that's present in this database. And it is using the software system that we used, it actually doesn't even include any of the MR data that is there. But if you look at the mineral corticoid receptor data, you can see that there are some sites that actually have higher levels of MR activity, like site 7, 8, and 9. One of the things that you can do with this data is try to correlate and see, does the activity of a particular receptor correlate with the chemical concentration. And this is, of course, it's maybe casual, it may not be causal, but there are certain correlations that make sense based on what you know about biological activity. And I'm showing you one example here with atrazine, looking at the atrazine concentration across the 9 different sites, and relate that to the aryl hydrocarbon receptor activity, and you can see that it has a very good correlation. You go back to the ToxCast data, and you can see that atrazine does in fact activate AHR. And this is something that was picked up in the ear approach as well. So there are some things, some correlations make sense. We've had a difficult time taking the known chemicals and trying to find correlations with the bioassays. For example, trying to predict which chemicals correlate with the mineral corticoid receptor. We're not finding many of the chemicals that were examined that have activity. And again, this is something that is kind of missing in the ToxCast database. So it would be a value, I think, of taking some of the chemicals that we know are present in the environment and do some additional dose response curves to generate data that we can then use to do not only to improve the ear approach, but also to try to understand some of the correlations that we have in these environmental samples between activity and presence. And again, I warned that I would show all the data that we generated for these samples with all the different assays that we've run, and that's actually on the right side, the right panel here. So this is all the different receptors that were looked at, the different molecular initiating events for all 80 of these samples. Again, just looking at the high to low values. And this is looking at what are the pathways that are affected. On the left side is the sum of all of the chemicals ears for a variety of different pathways. And I picked the top 50 values, the top 50 assays that have the highest ears. And then I grouped them based on their molecular initiating event. So you can see there are several assays that show increased activity of androgen receptor with these chemicals, increase constitutive androsten receptor, increase the estrogen receptor, and increase the pregnane X receptor. So these are the assays that were predicted to be active within the water samples. And then if you look at the real values, you can see that, yes, we're seeing the same thing. We're seeing increase in androgen receptor activity, estrogen receptor activity, CAR2 activity, as well as PXR. So in that case, the ear did predict the activity that we did actually validate within our assays. But it also suggested the ear approach also predicted that we might see FXR activity, glucocorticoid receptor, as well as activity of the PPARs. This was not evident in our assays, so it evaluated or added over concerns with these particular pathways that we were not able to detect. And again, as I mentioned before, the ear approach completely missed the MR, and the MR was something that we found active in all 9 sites. We had a high level of activity present at every site. We saw time-dependent changes that were there. Again, we would have missed that completely with the ear approach. We may be missing some other things too, and that abbreviated approach that I mentioned before, where we pool the receptors, but we couldn't look at the inverse agonism or the antagonism responses. Well, we may have missed some important activity there. So things like the estrogen receptor-related receptors, as well as the retinoic orphan receptor, these had a high level of activity within these samples. These would have effects on reproduction and inflammatory responses, for example, may have effects in wildlife species. So these are something that might be worthy of increased examination. And also, we have available to us some of these molecular initiating events that are that utilize the zebrafish genes instead of looking at the human genes. And you can see that we are getting some zebrafish assays that are active here, things like the androgen receptor as well as the aryl hydrocarbon receptor. And these types of data that are perhaps more environmentally relevant are not being interrogated to the same degree in the EAR approach. So to sum some of the positives and negatives of the EAR and the bioassay approach, the benefits of the EAR, this is straightforward. You're already measuring the chemical concentration. It's really quite simple to go ahead and try to see if you can prioritize which chemicals, prioritize sites. It uses the existing data. So again, this is something that you don't have to actually do in your own lab. You can do this just sitting at a computer. This is aimed at looking at additive effects and also takes into account mechanism of action, molecular initiating events tied to adverse outcomes. I find it very easy to interpret. Negatives, you can only look at the chemicals that you can measure and detect. So this is, in most cases, you are going in there with a list of chemicals that you want to analyze, and you start at that level. Even if you have a expansive list of chemicals that you can measure and you have measured, that doesn't mean that there is data present for you to do the EAR evaluation. And also, it doesn't mean that every assay is of the same quality. And I alluded to this with the androgen receptor activity, where there's one assay that tended to drive the EAR evaluation for one chemical. It turns out the same assay tends to over-interpret AR activity for a variety of different chemicals. Now, the bioassays have some benefits and negatives as well. The benefits, there are commercial sources for these bioassays. Every bioassay that I showed you in the previous slides are available from Indigo Biosciences. You can generate new data. You can look at receptors that people haven't looked at before. You can discover new receptors that are environmentally relevant. You don't have to depend on that data being deposited within ToxCast. And just like with the EAR approach, you're looking at additive effects and you're looking at the mechanism of action. And in this regard, you can also look at unknowns. You actually don't have to do any chemical analysis here if you don't want to. I think it adds to the overall project, but you certainly don't need to. The negatives, this takes resources and it takes expertise in order to develop this data. You have to be able to, get your extract into a cell culture compatible system. And then sometimes the interpretation is difficult, especially as you're looking across different receptors and you're comparing different sensitivities and robustness of response and looking at different reference compounds that are used for each of these different receptors. One thing that I came to conclude at the end of putting this talk together is really there are benefits to both assays, and perhaps the smartest approach would be to combine aspects of both. So you can use one assay, like use the EAR to generate a hypothesis for what chemicals or which pathways may be of importance. You can interpret and visualize the data using both of these assays, looking to use some of the types of bar graphs that are used in this EAR approach and use them for the bioassays. By using both, you're also generating the most amount of useful information. You are picking not only the predicted, but actually the measured response. So I think you have the highest quality data if you do use both approaches. And in this way, you can make better decisions. And again, what our goal is to make decisions based on the sites and chemicals and the bioactivities of concern within whatever our environmental matrix is. And with that, thank you for your attention, and I'd be happy to answer any questions that you have.
Well, thank you very much for that insightful presentation. As we move on to our Q&A session, I'd like to direct the audience's attention towards the handouts module within the GoToWebinar control panel, where they'll find some additional documentation relating to today's presentation to download for some further reading. I'd also like to invite the audience to continue sending their questions or comments right now using the questions window for this Q&A portion of the webinar. I've already received some questions for you, so we'll get ourselves started with those. The very first question that I have here states that AHR and PXR are activated by many environmental chemicals. So the question is, what might be the health consequences for this activation?
Yeah, this is a question that you get quite a bit because these receptors are both part of what's called the adaptive response that we have, where if we're exposed to a xenobiotic, a foreign chemical, we want to metabolize them and excrete them. And so these receptors have a biological niche where they're trying to induce drug and chemical metabolism. Unfortunately, this also leads to a change in hormone and vitamin levels as well. So how these fit into a pertinent adverse outcome is that these are changing levels of estrogen, androgen, and thyroid hormone, and then can lead to metabolic as well as endocrine disruption. From an environmental standpoint, we do know that activation of AHR in particular has consequences for reproduction in fish and other wildlife species.
Very good. Thank you so much for that answer. The next question here asks, have you tried predicting AC50 values for chemicals for which there are no ToxCast data using QSAR approaches?
Uh, we do have quite a bit of data on compounds using these exact same bioassays, but it hasn't been comprehensive. So it's been more on a kind of a hit and miss where we're trying to understand more with what's happening, and we may take a few environmentally relevant compounds and try to see if they are able to activate our assays. So things like the perfluorinated compounds, the PFASs, BPA, some phthalates. We have generated dose-response relationships for a variety of different endpoints. And then we're trying to use those to evaluate the sensitivity of our systems to try to look at these compounds as they're present in the environment. But I think it would make sense to be more comprehensive and try to fill in some of the deficiencies in the ToxCast database.
Very good. All right. Thank you. Next question here is wondering if the EAR approach examines all data in ToxCast.
It turns out that it doesn't evaluate all the different molecular initiating events and all of the different key events that are present there. And in some ways, that's a good thing. So it does tend to focus on nuclear receptors. It does tend to focus on particular enzymes that are known to have a signaling effect from environmental chemicals. But it doesn't look at everything. And like I said, in some ways it makes it easier to interpret, which is a plus of the EAR approach, but it does miss certain things, and it does miss things like mineralocorticoid receptor. I don't think it has a lot of progesterone receptor data in there, so it's just trying to be more simple. I think they have missed some things that maybe in later iterations they'll add back in.
Very good. All right. Well, thank you very much for that answer, for all of the answers here today. However, we have reached the end of the Q&A portion of our webinar. Now, if we couldn't attend to your questions, though, the team at Indigo Biosciences will follow up with you. Or if you have further questions, you can direct them to the email address that's up on your screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it will help us to improve our webinars. Now, I've also sent you a link in the chat box, and with this link, you'll be able to view the recording of this event on this page, and you can also share this link with your colleagues, which they can view once they register for the recording here as well. So I encourage you to do that. Now, please join me once more in thanking our speaker for his wonderful time here today. We hope you found the webinar informative. Have a great day, everyone, and thank you for coming.